My name is Nataliia Beztsinna, and I'm leading Crown Bio's ex vivo patient tissue platform. We are based in Leiden in the Netherlands, and where we focus on developing advanced ex vivo assays recapitulating human tumor microenvironment, which is also I will be covering today before handing off to my colleague Marten. If you followed our previous webinars, you are probably already familiar with this picture, recapitulating various events that happen within the cancer immunity cycle. And while my colleagues covered earlier other parts of the cycle, today we will focus specifically on tumor microenvironment, or TME. So what are the key players of the human TME? As you may well know, the main groups are immune cells, stromal cells, such as cancer-associated fibroblasts, endothelial cells, and of course tumor cells. The immune cells niche of the tumor is extremely complex, as almost all immune cell types, including macrophages, NK cells, DCs, T and B lymphocytes, can infiltrate cancer tissues. Importantly, it is not a binary anti-tumor or pro-tumor environment, but rather a mixture of these types. The overall phenotype is determined by the interactions of the immune cells with each other and with non-immune cells. The tumor and stromal cells play an essential role in determining the composition of the immune cells infiltrate, as well as the rate of recruitment and their in situ phenotype and function. It has been shown that for several cancer types such as melanoma, ovarian, colorectal, and renal cancer, the analysis of the TME composition can predict tumor aggressiveness and eventually patient treatment outcome. Several methods are established in the clinical and research practice to characterize the TME. Immunohistochemistry and immunofluorescence are used a lot, as they're relatively less expensive and preserve spatial organization of the tumor. However, the throughput of these techniques is quite low, and the number of markers is limited. Flow and mass cytometry allow higher throughput, higher number of markers with precise quantification, but lack the spatial architecture of the tumor. Newly emerging techniques such as RNA-seq or single-cell transcriptomics can be valuable alternatives to the classical methods, offering high throughput and in-depth characterization of the TME. Here you can appreciate an example of a transcriptomics tool known as microenvironment cell population counter, or MCP counter, developed to characterize the tumors. MCP counter relies on the analysis of transcriptomics markers that are only expressed in one cell population and provides scores that are proportional to the cell numbers in the analyzed sample. Signatures are available for 8 immune cell populations, as well as for endothelial cells and fibroblasts. This approach is robust and allows to compare different samples. Based on the abundance of adaptive and innate immune cells, the tumor types in this heat map were classified as hot with relatively high abundance or cold with relatively low abundance of immune cells in the TME. Interestingly, the number of somatic mutations in the tumor cells does not always correlate with the degree of its infiltration by the immune cells. Another example from our own data showing differences in the immune TME composition between different samples of the same indication, non-small cell lung cancer. We analyzed the composition of malignant pleural effusions and surgical resections of the several patients by fax and found that compared to resected material, malignant pleural effusions had relatively higher percentage of immune cells and in particular CD3, CD4 positive. Identifying such differences from one indication to the other and from patient to patient within the same indication is not only important for the predictive value of the human TME analysis, but also for selection of the right contexture for the novel therapies testing. You might have seen these graphics in our previous webinars, but I would like to show it to remind again how in vitro assays can recapitulate the human TME. We can either focus on the reconstituted models, where each cell subtype is selected and combined in a LEGO-like approach. Or we can test the therapies in a native TME assay that gives a unique footprint of each patient's tumor. In a previous webinar, we asked you a poll question about which types of the assays are you performing with native TME. Here are the responses that we've got from you. 37% of the responders have been using the native TME assays in the past, with the majority focusing on tissue slices and 3D ex vivo assays. Today, I will show you some examples of such assays from the literature, as well as from our own research. This paper by Marten and al. Shows an interesting example of using fresh tissue slices from liver metastases of colorectal cancer patients. The researchers used a device called Vibrotome to generate precision cut slices of 100 to 200 micrometer thickness. Before cutting, the tissues were embedded in agarose gel. The authors have shown that the slices were viable for at least 72 hours in culture, and then they closely recapitulated the original human tumor biology, as shown on the pictures here of two examples of patient 3 and 4. Several treatments, including immune checkpoint inhibitors, were applied to the cultured slices, and the authors assessed treatment responses by quantification of Ki-67 staining with immunohistochemistry and by scoring different areas with the slices representing tumor, stroma, and necrosis. They have found that 2 out of 9 tested samples mildly responded to immune checkpoint inhibitor treatment, and the examples of the response data from these 2 samples are displayed on this slide. Another interesting approach is ex vivo explants. It's presented here from a paper of Kokoninos et al. The authors used pancreatic cancer explants of 1 to 2 mm in diameter that were cultured on a gelatin sponge. The culture was much longer than in the previous example, up to 12 days. The authors showed that the explants maintained the morphology of the original tissue on day 5. Some of the explants were exposed to an anti-cancer drug Abraxane and have shown sensitivity to the drug by a tonal assay. At Crown Bio, we have our own method for performing native TME assays. It is called ex vivo patient tissue platform. We process fresh tumor samples from resected material as well as from ascites pleural fluids and generate tumor clusters of around 100 to 150 micrometers by gentle mechanical shearing. The clusters are then mixed with a defined hydrogel matrix and seeded in 384 well plate at a density of 50 to 300 clusters per well. The cultures can be then exposed to various drugs and combinations, and the drug effects are measured upon using our proprietary phenotypic image analysis platform. Histological analysis of these 3D ex vivo cultures has been shown to closely represent the original patient tissue. We have tested the sensitivity of non-small cell lung cancer tissues to immune checkpoint inhibitors and immunostimulatory controls in our EVPT assay, what you can appreciate here on a heat map. We have found that around 20% of tissues respond to immune checkpoint inhibitors, and this is correlating with the clinical data for this cancer type. If we take a closer look at one of the responder tissues, we can see a highly diverse sample consisting of tumor clusters and supporting cells in a non-treatment control. The treatment with immunostimulatory control superantigen A drastically reduces number and size of the tumor clusters while increasing numbers of proliferating single immune cells. Similarly, but less drastic response was observed with ipilimumab treatment. Our image analysis specialists have developed a method to analyze these phenotypic changes in a response to immunostimulatory treatment and dissect tumor-killing responses from increased immune cell counts. The methodology is based on size and shape of the cells and not employing any specific biomarkers, therefore allowing for a higher throughput of the analysis. Such phenotypic analysis is translated into the following data. This sample, for example, have shown a response to SEA and a mild response to CD3/CD28 beads and pembrolizumab. It has also shown increased immune cell counts upon treatment with SEA and CD3/CD28 beads. Our measurements in functional readouts in the EVPT assays were confirmed by multiple cytokine measurements done on the same sample. Here again we see an increase in interferon gamma measurement, granzyme B and IL-2 upon treatment with SEA beads and pembrolizumab compared to the median control. In addition of using native TME of a sample that might or might not contain the infiltrating immune cells, we can add autologous PBMCs or other immune cells isolated from the blood of the same patient. We call this assay enhanced TME as it provides additional measurements such as immune cell migration and infiltration. These are possible because of the PBMCs pre-stained with the cell tracker before being added into the assay, and therefore their spatial organization can be measured. This heat map shows non-small cell lung cancer patient tissue sensitivity to drugs in co-culture with autologous PBMCs. As this is relatively new setup, the tested number of patients is smaller here, but some interesting effects could be observed, such as when the sample is sensitive to SEA, it has a higher chance to be sensitive to other immunostimulatory treatments. One of the examples is showing almost complete tumor killing upon treatment with the immunostimulatory controls such as SEA and CD3/CD28 beads and immune checkpoint inhibitors ipilimumab and pembrolizumab. On the images you can appreciate here almost complete disappearance of the tumor clusters and increase in numbers in the PBMC counts. As I'm reaching the end of this talk, I would like to show you these images from our research highlighting the differences between fresh ex vivo cultures and the patient-derived organoids. On the left, you can see an image of a suspension generated during our tissue processing protocol. It contains tumor clusters of, as I mentioned before, 100 to 100 micrometers in size and a lot of released supporting cells. When the sample was seeded in a hydrogel in our EVPT assay, we can see a rich representation of the TME, tumor clusters, elongated cells similar to cancer-associated fibroblasts, and multiple single immune cells. If we continue to culture the exact same sample from multiple passages, and here at the passage 6, we reach the state where all other cells have disappeared and only patient-derived organoids stayed behind. With this, I hope I provided you enough insight about the various possibilities of the ex vivo assays with native tumor microenvironment. I would like to conclude by listing the main advantages of these assays. such as close recapitulation of the human TME complexity. The speed of the assay, we don't need any pre-culture time and can work straight from the patient. The recapitulation of biological variability between patients. Think of the in vitro clinical trials. Relatively easy to establish these assays. Among the challenges, we can list the scalability of the model, limited data available before the assay on the patient, and the difficulties with patient tissue access and sometimes costs. Now I will hand over the microphone to my colleague Marten, who will present you the reconstituted TME assays. Thank you very much for listening, and I'll be happy to answer any questions.
Thank you, Nataliia, for your very nice talk. And now I will continue with discussing the suppressive TME and especially why cellular context matters for immune and drug responses. So why do multicellular preclinical models help to accurately predict drug responses? Envision cancer. Cancer is essentially just unwanted cellular behavior, but unwanted cancerous cellular behavior you can see as purely pathological molecular signaling. And then on top of that, molecular signaling is highly dependent on the context. So if we study cancer and drug effects in the right context, we will get better predictions for effective drugs, which means shorter timelines and lower cost waste for drug discovery and implementation, and most importantly, happy stakeholders, which are, of course, the patients. So, how does a signal go from context to effect? If we envision a textbook example of cancer cells, we see one cell that has either a signal that it receives or a mutation that drives a certain signal, which will be translated into a gene expression profile and then elicits an effect. But it is sadly not that simple because these effects are often modulated by either signals that are operating in parallel, the tissue in which the cell is present. So, for example, this epithelial cell is connected to other epithelial cells, which have apical basal and planar cell polarity, which determine for the cells what's left and right and what's up and down. Of course, an effect can only be reached when the building blocks are available. So the metabolic state will highly affect this signal. And then we have some additional external signals, of which one very important one is the extracellular matrix, or the anchorage of cells to the extracellular matrix is essential, especially for survival signals. And then there's many other cellular components like cancer-associated fibroblasts, here illustrated as a cuff, which can either secrete cytokines and growth factors or have direct integrin-mediated connections to the cancer cells. And on top of this, we have immune cell populations, importantly the myeloid population, which can affect the tumor cells directly, but are also having a lot of crosstalk between themselves and the fibroblasts, and of course the cell types that we have been mainly discussing in these webinars, namely the lymphoid cells, which can be affected by either the CAFs, the myeloid cells, the epithelial cells, the cancer cells, and of course themselves. So in this talk, I will mainly focus on the three cell types on the right, so the cancer-associated fibroblasts, the myeloid cells, and how to model these in vitro to study both cancer drug development and lymphoid activity in the sense of immunotherapies. So first, we have to go over a few model basics, especially for the people that are jumping in for the first time in this seminar series. So in this case, I wanted to go over the relevance and context of the different preclinical assays that are currently widely used, which have a nice balance between how scalable and cheap they are to how physiological relevant they are. So 2D cell cultures, almost everybody is aware of those. These are monocultures of cancer cells or other cells in 2D, which are very easy to work with, very scalable, but of course lack all context and physiological relevant environment and stimuli to actually model the tumor microenvironment. Then if we go a bit more to the physiological relevant side of things, we have spheroid assays, so 3D. Already, this can be performed with 2D cell line cultures. You can culture them in 3D, hereby easily adding at least another spatial dimension to your cultures. But today, I want to focus mainly on the organoids, which are primary tissue isolated cancer cells and are growing in 3D. And of course, Nataliia already covered very nicely the ex vivo patient tissues. So I want to, in this talk, take a step back from the ex vivo part and focus on how we can use organoids in a Lego-like fashion to model the diseases that we are interested in and introducing different kinds of contact and cells in these cultures. So first, a quick recap on what are organoids. So you can envision this as a living biobank of tumor models that are coming straight from the patient. So these can be normal or tumor or tissues. And when isolated from the patient, these reflect the genomic, morphological, and physiological characteristic of the corresponding tumor in vivo. So organoids are developed directly from adult stem cells from patient tissues. So like what you see here on the left side, so either you select from, you grow your organoids from a biopsy, or that you first go from a biopsy into the mouse and then towards organoid cultures from those tumors that arise here. So both have upsides and downsides. So in this case, we don't use the mice models to expand the organoid models, but that so this comes with the benefit of quick isolation and growth. But then using these mice models is actually also very beneficial, because the moment that the biopsy hits the mouse, actually even better recapitulates the tumor of origin, and these isolated organoids already are well validated to use in vivo for if you want to use the same organoid lines for in vivo follow-up. So both come with, but this is a more elaborate and more expensive procedure. So this is really depending on the purpose that you want to use these organoids for. And as you can see, there's a large list of already available cancer indications for which organoids are available in both PDX and PDO setups. So the organoid view on cancer is of course still very limited. Because here this is actually how an organoid would look if we culture them as well. So it's a nice monolayer epithelial-like cancer cells. But of course, if you look at the whole tumor, these organoids are actually only these cancer cells are only a very small fraction of the total tumor volume. So this is a nice example of a pancreatic cancer, which is notoriously well known for their large stromal contribution, so the big stromal compartment. These important cells in this stromal compartment that I want to focus on today are myeloid cells and fibroblasts. So myeloid cell populations consist of monocytes, macrophages of M1 and M2 types. Dendritic cells, these are examples of myeloid cells that are important to study cancer. And then, of course, the fibroblasts, known also as cancer-associated fibroblasts. I will discuss all these four cell types in more detail during this seminar. First, the role of myeloid cells in cancer. Especially macrophages have been notoriously known to be involved in tumor progression. There are different polarizations of this subtype. So we have M1 macrophages, an in-between phenotype, and M2 macrophages. The M2-like macrophages are also known as tumor-associated macrophages and are known to have pro-tumorigenic effects like immune suppression, stimulating angiogenesis, facilitating tumor growth. Then on the other side, we have the polarization phenotype that we call M1. which is a anti-tumor type, or at least with anti-tumor activity associated macrophage type, which is pro-inflammatory, stimulates antigen presentation and T-cell activation. So obviously, these are populations that we want to modify in cancer therapies. There are various ways to jump into these strategies. So, for example, you can either inhibit macrophage recruitment, lowering the amount of tumor-associated macrophages. You can specifically target tumor-associated macrophages by trying to kill them. You can also try to reprogram tumor-associated macrophages from M2 to M1 macrophage phenotype, or interfere with don't eat me signals that make cancer cells more vulnerable to immunotherapy. So how would we model and study different types of myeloid cells? And this is actually very nice thing to make use of the different sizes and morphologies that they have. So here I already illustrated it, that M1 macrophages are more round and flat, M2 macrophages are more spiky, are less round, and then the dendritic cells are even more spiky. And this has actually already been used in literature to start differentiating between the M1 and 2-like states, just purely based on imaging and morphological analysis, or things like the axis and the roundness. All these parameters can be used to model either one or the other. And this is actually what you see here very nicely. So the M2-like macrophages are very nice spiky small cells, and the M1-like macrophages are round, large cells. And in this study, they also showed already that based on these morphological features, by playing around with different cancer therapies, especially the CSF1R inhibitors and TLR simulation, are effective in resulting in M1 enrichment. So you can actually modify the macrophages and affect their polarization. In our own R&D, we wanted to really put this more in 3D. So you can imagine that in 2D cell lines, the dimension that you can characterize phenotypic properties are limited to that dimension. So in 3D, we would expect that this is even better to model, and this is indeed what we see. So we can find that in 3D cultures, myeloid cells recapitulate morphological features as seen in vivo. So what we do in these procedures is we have PBMCs, we isolate monocytes, cultivate them in 3D, and stimulate them with different cytokines to grow them into different polarization states. And this is actually how that looks. So if we use our in-house image analysis software, we can identify distinguishing phenotypic characteristics. So here you have the monocyte, very small round cell, M1 macrophage, a larger, rounder cell, an M2 macrophage, even larger with more protrusions, and then dendritic cells, which are very large and have even more larger protrusions. So what we did is subsequently we made a classifier and trained our image analysis software using the shape, the size, the spikiness to get phenotypic profiles for each cell population. And this actually works very well. So we have here a multi-feature score and the size. And then we see that in this space, we can discriminate the different populations of myeloid cells. So if we then start looking at drug responses in our 3D setups, we see that we can go from M2 macrophage polarization to M1 macrophage polarization by adding different treatments like the STING agonists or CSF1R inhibitors. This is really, you can clearly see a shift in the space towards M1 phenotype from the M2. So this is working really well. This is not only our morphological features are in line with this, but also the, for example, excretion of IL-10. We can see that this also is corresponding to more M1-like macrophage behavior. So now we are talking about monocultures, but if we are looking at co-cultures, we can actually see also that myeloid cells are affected by the presence of tumor cells. So this is an example of tumor cells grown in 3D combined with M1 macrophages. And zoom in, and when we start quantifying these feature spaces, what we see is that we can still distinguish the different populations of myeloid cells when there is no treatment. So if they are in monoculture, and then when we start adding tumor cells, we can actually see that the CD14 positive monocyte population is shifting to a more M2-like state, and that these two M2 and M1-like states are more moving to a more general in-between state between M1 and M2. So by just adding culture, by adding the different cells together, we can already start modulating the polarization state of these cultures. Then we have co-cultures. What you can see is also that this is different, changing depending on if the cells are close or not to the tumoroid cells. So for example, the M1s are infiltrating into the tumor while the monocytes are located more distantly. And this is a final example for the myeloid cells I want to use as an example. So here what you can see is that when we have naive CD8 T cells or pre-activated CD8 T cells, we see very high tumor cell infiltration, but as soon as we start adding monocytes, we see reduced infiltration, and when we have compounds that interfere with these monocyte activities, we see that the infiltration again increases. So it is possible to modulate the activities of these cell types with drugs in vitro. Okay. Then I want to wrap up my talk by just going to my personal favorites. So we have fibroblasts also present in tumors, and they are central in cancer cell behavior and drug responses. We discriminate nowadays 3 different populations of fibroblasts, which are inflammatory calves, which are important for immune suppression, cytokine excretion, and they do have seem to have a tumor promoting role. Then we have myofibroblastic calves, which form the fibrotic part of the tumors. They can also both interfere and stimulate tumor progression, and the newest one is the antigen-presenting calves, which have been found to be able to cooperate with the immune system and modulate immune cell responses as well. So it is important to be able to model these type of cells as well. And there has been quite some effort in that domain in academic settings. So we already know that fibroblasts can provide a stem cell supportive niche, especially in pancreatic cancer. We know that CAFs are increasing chemo and radio resistance, stimulate mesenchymal features and metastasis by being involved in epithelial to mesenchymal transition. They are published to show a physical interaction with tumor cells, and this also contributes to their tumor growth. And we have now already multiple examples of autologous co-cultures between calf cancer cells and immune cells, so triple co-cultures that are able to study the interaction between the different compartments of a tumor. So this is a very important example that I want to give, and it really shows that context matters. So for example, PDAC, pancreatic cancer subtype, that is normally very mesenchymal and doesn't contain a very clear LGR5 or OLFM4 stem cell population. If you start looking at different ways of culturing cancer cells and co-cultures, so here we have organoid monocultures, organoid co-cultures with fibroblasts, but then they are just homogeneously added to the gel, and then a mini tumor type co-culture where the cells are first aggregated before plated, and so this looks a bit like these. What you then see is that markers that are so mesenchymal markers, the first 5, and stem cell markers only change in the context and become more physiological relevant in a context where they really touch each other. So context really matters if you want to recapitulate tumor. in vivo. So, of course, we are trying a lot in this domain to get these co-cultures also ready for preclinical studies. So one of the setups that we have been developing is our co-cultures between tumor organoids and fibroblasts. And this is an example of how we run this type of assays. So we have tumor organoids and fibroblasts that are homogeneously seated in hydrogels. We let them recover for a few days so that the tumor cells and fibroblasts can grow out. And then we add a compound of interest. In this case, this example uses cisplatin. And after a set amount of days, we fix and stain these co-cultures and quantify the effects. And what we clearly see already in these kind of assays, so here we have a organoid only or an organoid fibroblast co-culture, that in the presence of fibroblast, tumor cells start growing more efficiently, so the tumoroids are larger in general, and also that resistance, in this case to cisplatin, is increased in co-cultures of organoids with fibroblasts. So what you can see here is that these 1 in 10 doses are quite a bit higher than if you don't have any fibroblasts there. So we can recapitulate tumor supporting effects of fibroblasts in vitro. Then to go to the next step is we have an automated assembly of different types of co-cultures with pre-activated PBMCs. So again, we seed tumoroids and fibroblasts, but then add PBMCs on top. And when we then characterize these after a few days, we can really score all kinds of parameters, including infiltration to killing, etc. So this looks like this. So we have either organoids only, fibroblasts only, the double co-culture between fibroblasts and organoids, and then we add on top PBMCs or PBMCs that are activated by superantigen SEA. And below you can see the segmentation masks that our image analysis software is making to make the final quantifications of these different compartments in these cultures. And on the right side, you can see a quantification of tumoroid killing in response to SEA. So if you have no PBMCs or SEA, or we add naive PBMCs on top of that, we don't see much killing. But then if you have super antigen present in these cultures, you can see very effective killing in these setups. And then I want to wrap up. We are still pushing the development. As I said, physical interaction between tumoroids and fibroblasts is essential to get relevant expression of your in vivo, of your pathological relevant expression of your markers. So we are trying to push that by making mini tumors. So we're trying to set up a preclinical setup where we have high throughput capabilities to study these mini tumors. And if we zoom in on the right side, we can have already quite a nice example of tumor organoids that have been cultured with PBMCs, and we can quantify infiltration into these mini tumors and killing when we stimulate with different drugs or super antigen. And that's where I want to leave it for today. I hope I convinced you that there are already multiple flexible setups that we can use for different immune oncology approaches, and also to help us with studying the cancer immunity cycle, especially the suppressive part of this cancer immunity cycle. I provided now an example where we use organoids in combination with quite a variety of myeloid cells and lymphoid cells, as I said, like small Lego blocks to reconstitute the tumor of interest with the cell lines of interest, and then use these co-cultures to make all kinds of measurements on killing, infiltration, polarization, and hopefully, this will help with discovering some very interesting therapies in the near future. And with that, I would like to summarize up all the different aspects that we have been covering in the cancer immunity cycle seminars, also the first and the second session of a few weeks ago. And of course, if you have any questions, reach out to us. We are happy to discuss.